Hi everybody, today we're going to talk about metagenome sequencing and how you process metagenomic results. So we've talked about 16S sequencing a fair bit in this class so far, and we talked about some different ways that you can plot results from 16S ribosomal RNA gene amplicon sequencing experiments, which again give you an idea of who is there, whereas metagenome sequencing gives you an idea of what they might be doing because it gives you access to information about all of the genes instead of just this one gene that we sequence all over and over and over again in a 16S sequencing experiment. And we talked about phylogenetic trees last time and how that's one useful way to get results from 16S sequences. So now today in this video, I'm going to focus on metagenome sequencing. And I mentioned before, a first look at a metagenomic data set could be a simple categorization of which general metabolic functions the gene seems to be likely to be participating in. So for example, you could list how many genes you found in your data set that seem to be associated with photosynthesis, or how many might be capable of oxidizing hydrogen or other molecules, or it looks like it has the methanogenesis pathway, or it has a gene that's predicted to be able to allow the transport of iron, or you don't know what the function of that gene is because it doesn't have any results in metabolic pathway databases. And so today what I'm going to talk about is, okay, how exactly do you get to this point? How do you make one of these determinations that a gene is involved in one pathway or another? So that's what I'm going to talk about today. First, a little bit more about how you generate the metagenomic data in the first place. So here I've got a bucket of water representing my environmental sample. So this could be a bucket of water from the ocean or from a lake or from a river, or you could also think of it as a sample of sediment or of soil or a fecal sample or any sample you want. We extract DNA from that sample, and now when we prepare that DNA for metagenome sequencing, we have a couple of decisions to make. We can decide how large the chunks of DNA are that we want to sequence, and we can decide what is our strategy for sequencing. What I've drawn here is a strategy that's called paired end sequencing, where each one of these solid bars represents a sequence that is paired to another sequence over some gap. Okay, the gap represented by the dotted lines. And so if we chose to fragment our DNA into bases of, let's say, 500 bases, and we sequence 150 bases on one end and 150 bases on the other end, we now have covered a total of 300 bases out of the 500 bases on our chunk of DNA. So we've got a gap in the middle of about 200 bases. Right? So 150 and 150 makes 300. We've got a gap in the middle of 200. So now we have covered, that is how our 500 bases of genomic DNA is distributed. Now, when we fragment the DNA, that's a somewhat messy procedure in the lab, and it has some error. So we don't know exactly how long the chunks are. We just know that when we ran our gel in the lab, and we selected fragments that were approximately 500 bases, but there's going to be some wiggle room there, and so we don't know exactly how big this gap is between the sequences. But if we did our lab work carefully, so this is an example of how lab work directly translates into good data at the computational stage. If we did the lab work carefully, we know that the gap in the middle is approximately 300 bases. And that can be really useful information for generating the assembly. So now we're at the stage where we have our data from the sequencing center, we're sitting in front of our computer, and we have a file that's many gigabytes large, and we have to figure out what to do with it. And one of the most important things we could do with it is to assemble it into larger chunks of DNA. Instead of just individual 150 base pair sequences that are paired with other 150 base pair sequences over some gap, we feed all of this data to an assembly software program that puts them together. So here I've drawn these red bars to represent assemblies of these red individual sequences. So the assembler noticed that these sequences that are red overlap with each other and build a network of an interconnected graph where they have a series of overlaps that connect them to each other. And you can simplify that with putting them together into one assembly. So from these individual sequences, you get a hopefully shorter list of assemblies. The technical term for these individual chunks of an assembly are contigs, which is short for contiguous assembled sequence. So the product of an assembly in a metagenomic experiment is a series of contigs. Okay, so now we have our contigs, and drawn here are this is another way of drawing an assembly. So here we have individual sequences. Here we've assembled them into contigs. Here the red contig is composed of 4 individual sequences, perhaps represented by 2 pairs of 2 sequences each. Here is a green contig represented by several different individual sequences. Here it's not clear which ones are paired or not. It's just a PowerPoint figure. And here's a blue contig represented by 2 individual sequences that were assembled together to make the blue contig. Okay, so a product of an assembly is contigs. The contigs should contain 1 or more genes. If it is a really good assembly, you'll have very long contigs that might even represent an entire genome. Usually it's going to be some reasonable fraction of a genome. So within each contig, we can predict genes. So here's an example of a start codon followed by an open reading frame that encodes a recognizable predicted amino acid sequence. So here's one gene encoding one protein that is found on one contig. One contig could have multiple genes encoding proteins, and different contigs will have different genes and contigs, right? The contigs are chunks of different genomes. The contigs could be from the same genome, and that we just didn't have enough data to be able to link them together into the same contig/genome, or more likely, each contig is from a different species. So it represents a fraction of a genome of a different species. But without more work, you actually don't know that yet. All right, so the point of today's lecture, we could spend a whole course on different assembly methods and different gene prediction methods and so on. But what I wanted to focus on today is if you get to the point where you have a bunch of predicted protein sequences from your metagenomic assembly, what you really want to know is, okay, what do my proteins do? All right, so we're fast forwarding through all these steps where we're cleaning up the data, we're assembling, we're assembling the data, and we're predicting proteins, and now we're to the point where we have a big list of proteins that our metagenome encodes. Okay, so the point that I'm going to focus on right now is, what do you do with those predicted protein sequences? And the answer to that is you compare them to a database, and usually compare them to multiple databases. And so in this simplistic example I've got on the PowerPoint slide, we have this protein sequence encoded by our gene in our metagenome. We submitted that as a query to a database, and in that database it found a protein sequence that was very similar, although not identical, to an entry in the database. And so we can say that in the database, if this protein is known to be associated with a certain metabolic pathway, we're now reasonably confident that our protein is also associated with that same metabolic pathway. Okay? Now, that's all we can say, is that we are reasonably certain, or that we can say we are uncertain, because all we can do at this stage is really to compare our sequence to a database of known sequences. In a lot of the projects that I work on, and many people work on, the most common answer is yes, there is a match in the database to your predicted protein sequence. It has a reasonable match in the database, and the function of that protein in the database is not known, right? So in order to actually generate new knowledge about the functions of proteins, that requires a lot of lab work, a lot of actual microbiology and genetics and biochemistry to actually work out the functions of proteins. And if nobody has actually done a whole PhD project on your protein yet, then there probably isn't any information on it in a database. And in microbiology, because we've studied only a tiny, tiny fraction of microbial diversity in the lab, it's very common that you will find a match in the database to other metagenomes that have proteins that do not have known functions. Okay, so that is nevertheless still one of the most useful things we can do with the metagenome. And then that's just the first step with this kind of analysis. The next step is even if the predicted functions are not known, we can still get more information, especially if the whole project was designed in such a way that is conducive to getting more information. So one example of another layer of information we can get is the abundance of each predicted gene and its encoded protein among the assembly. We get the abundance in the environment. This is a true environmental abundance with some caveats. The abundance comes from the number of individual reads that were mapped against that contig. So that's why I started out by talking about how these contigs are generated from an assembly of individual reads, because at the end of the analysis, we make use of those individual reads again as a way of counting the abundance of the proteins that are encoded by that metagenome. Right? So the way this works is that we have our list of proteins. We take all of our individual reads, map them. Read is another word for individual sequence. So a read is one sequence. We map the reads against the assemblies, and we can say that this gene here encodes a protein, and it's the abundance of that gene in the environment is represented by the number of reads that are mapped to it. I'll talk about the numbers in a second. Right? Maybe I'll just do that right now. It probably makes most sense as a comparison to another sample. So what's most useful is to not just do this with one sample, but to do it with multiple samples. So here we have two samples, a green sample and a blue sample. And for this particular gene, it has a greater abundance in sample 2 than in sample 1. You can also say a library. Library is a more technical term in the sequencing world to represent the actual collection of DNA that you gave to the sequencing center, because at that point you're actually a few steps removed from the sample. Nevertheless, in practice, we often just say sample because it's more common to say sample than library. But library is the thing that you make from the sample. And library 2 or sample 2, if you count how many at each position, on average, how many blue reads are lined up at that position. So here we have 1, 2, 3 at this position. At this position we have 1, 2 actually only. At this one we have 1, 2, 3. At this position we have 1, 2, 3. At this position 1, 2, 3, 4. At this position, right? On average, about 3 or maybe 4x, 3 or 4 reads are lined up at each position of this gene. Whereas with library 1, we only have 1 or maybe 2, and in rare cases, 3 reads lined up at that position. So in general, more blue reads were mapped to this gene than green reads, so we can say this gene was more abundant in sample 2 than in sample 1. Right? Even though this is a silly, simple example, you can see how powerful that could be. So if we did an experiment where we sequenced 100 fecal samples from 100 different individuals, we could then tell you which genes were more or less abundant in each of those individuals. Right? Not just that they were there, but actually their abundance. And again, this is metagenomic sequencing, so we're talking about all of the genes, not just the 16S gene. So we can say not only which species were more or less abundant in some individuals than others, or in some soil samples or in some water samples than others, we can also say which metabolic pathways were predicted to have been encoded and which individuals or in which samples they were more or less abundant in. Right? And even for those genes that we cannot assign a function, we can still say this gene that has no known function is super abundant in these individuals and not abundant in those individuals. And that could be a really important clue for trying to figure out what its function is. So if it's only abundant in individuals who have a disease, for example, that is at least a potential clue worth following up on. It's almost never enough information to actually prove anything, but if you need any angle at all to get started on your project, that can be a really useful starting point. Okay, so that's how you get abundances from contigs, which again are assembled from metagenomic sequences. All right, so another layer of analysis that you can layer on top of the assembly stage is to bin the contigs. All right, so before I was talking about this stage where we have assembled contigs and we can look at the proteins that are encoded by those assembled contigs. There's another thing we can do, which is to bin those contigs into what are called metagenome assembled genomes. Or colloquially, we just call them bins because it's a binning process that makes them. So these are basically genomes. So in metagenomics, we are very rarely able to produce a 100% complete assembly that represents 100% of a genome. That's just the nature of metagenomics. Nothing is ever perfect. But what we can do is use various computational tricks to take all of the contigs that seem like they probably belong to the same genome and put them into one bin. Okay? So we can say that these collection of contigs, they share sequence composition, they share certain properties, they match up to certain databases, they all seem to go together, they may have reads that are paired with each other. So we think that these all belong to the same genome, and we call them one MAG, metagenome assembled genome, or one bin. So it's a genome that has been assembled entirely with metagenomic techniques, not by growing a pure species in the lab and sequencing its genome. And it's represented by multiple contigs still. You never have one single contig representing the whole genome in this case, unless you do more work. But you have hopefully a relatively short list of contigs representing each of these bins. So now all of the analysis that we did with the proteins in the assembled contigs, we can now do on a bin by bin basis. So we can break down these results like the predicted metabolic pathways and the abundances of the proteins in each of the individual samples. We can now report those results instead of for the whole assembly, which represents all of the genes in the whole sample. We can now make those results for each individual bin. And we can say that the, let's say the methanogenesis genes in this genome, or this species, because these different genomes almost certainly represent different species, the methanogenesis pathways of this genome are very abundant in certain individuals, and methanogenesis genes, they're the same genes, it's the same pathway, but they are specific to a different genome, are abundant in different individuals. Right? So on a species-specific level, we can look at differences in abundances of individual proteins, even if those proteins essentially have the same name or they belong to the same pathway. Right? It's just a long way of saying that we can report these results on the level of the whole assembly, which is the whole community from that sample, or by doing binning, we can report these results for each on a species-specific basis. And you can probably appreciate how that could be a lot more valuable than just saying that these genes are encoded by somebody in this community of microbes. Now we can say that this gene is in fact encoded by this one species and not that species, or it is encoded by that species, right? So you can work at the species level instead of the whole community level, even with metagenomics, and even though we still haven't grown anything in the lab. So here's another slide showing much of the same information that I explained before, but it's drawn by someone who's a little more skilled with illustrations than I am. So I thought it'd be useful to kind of go over the same material again as a review of everything I said today with an illustration that's perhaps more pleasing to the eye. So here we have our mixed community of microbes. We extracted DNA from our mixed community, so now we have a pool of DNA molecules in the lab. We do metagenomic sequencing. Shotgun metagenomic sequencing just means random sequencing of all of the DNA. What I call true metagenomics is shotgun metagenomics. We have our reads generated from those DNA molecules, but here it's turning into black and white to acknowledge that we actually don't know which read comes from which DNA molecule. And that's a trick that we will use later on. So within assembly, we can turn these individual reads into contigs. I talked about that already. And then we can generate information about these contigs, including the coverage. Coverage is the technical term for abundance, because the way that you calculate abundance is by lining up the reads along the assembly and counting how many reads cover each position of the contig. So that is the coverage of the reads against the contig. That's how you get information. And here it's highlighting that you can do this with one sample, but it's a lot more powerful if you have lots of samples. I showed an example with 2 samples, And I described a hypothetical example where you had 100 samples. The more samples you have, the more information you have, because you can make use of the differential coverage of each gene or each contig in all of your samples. So that's a really important layer of information that you have about your sample. Another layer of information you can have is the kind of statistical sequence composition. So here's an example where you're using 4 base sequences to generate just a statistical table showing which contigs have similar inventories, similar compositions of 4 base sequences than others. And you use both this information, this computational statistical information that has actually no biological content as well. This is just like a computational data science information theory kind of thing that I'm not really going to talk about in this class. You just feed it into a computer program and it does it for you. I can talk about it if you're interested. But it is good to note that there are 2 basic kinds of information we can use to do the binning, which is the next step, is we take the coverage information, which is to say the abundance of each of the contigs in each of your samples, this computational statistical information about the composition of the A's, C's, T's, and G's in each of the contigs. You feed that into a computer program and it takes the contigs and bins them. And this is where it's often much more useful to start generating results, as I mentioned on the previous slide, because you now know that these contigs were most likely generated from the same original DNA molecule, the same DNA genome, and should be considered to be part of the same genome. These were originally coming from the same genome, and now we know by doing all this work that they probably belong to the same genome. These techniques make mistakes, but there have been lots of studies now to show that they're actually amazingly accurate when used appropriately. So the point of these colors here is highlighting that at these intermediate stages of the analysis, we don't know which genome each sequence belongs to. They're just essentially random collections of sequences. And by doing all this work, like looking at the coverage of each contig in different samples and looking at statistical information about the contigs, we can group them into bins that should represent different species. And by analyzing these bins further, we will eventually realize which species they actually belong to and what genes they encode. Okay, that is my overview of metagenomic assembly, metagenomic sequencing. And we're going to talk about these concepts and play with them and actually do real things with them together in class. And I look forward to doing that with you in class. See you then.